Hello, my name is Kara Gardner. I'm currently a senior in the Hazen Laboratory studying plant regulatory genomics in the model grass species Brachypodium distachyon. One of the reasons we care about plant regulatory genomics is because of plant cell walls and their relation to anthropogenic climate change. So to start off, plants serve as a massive carbon sink. Plants fix atmospheric carbon dioxide and incorporate the carbon into the main components of their secondary cell walls, which are cellulose, hemicellulose, and lignin. Thus, a better understanding of when plants incorporate carbon into their cell walls can introduce novel strategies on how to mitigate impending repercussions of climate change. So to study secondary cell walls, we're using the model grass species Brachypodium distachyon. B. distachyon is a model organism for cereal crops due to its short life cycle, simple growth requirements, phylogenetic relation of food crops, and ease of transformation. And very briefly, I would like to point out some structural information about brachy important to know in this talk. So spikelets outlined in blue are where the flowers develop. Nodes circled in red are where new cells and stem growth are generated. Internodes shown in the white brackets contain cells from the stem that may be actively growing and elongating, or cells that have finished elongating and are now thickening their secondary cell wall. On the right side of the slide, we have an example plant cell showing a very general structure of plant cell walls. Unlike mammalian cells, plant cells have cell walls that give the plant structural support, protection from pests and pathogens, and allow the plant to resist forces associated with water transport. Primary cell walls are tensile and are synthesized during plant elongation, while secondary cell walls are very rigid and are synthesized in certain cell types after the cells have stopped elongating. So going back to the previous slide, we can recall that these cell walls contain a biomolecule called cellulose. Cellulose makes up the majority of the cell wall in grasses and is a polysaccharide consisting exclusively of long chains of 1,4-beta-linked glucose that associate laterally into a mycofibril. Within the plasma membrane, the structure synthesized in cellulose is known as the terminal complex and resembles the hexagonal rosette composed of cellulose synthase A proteins, otherwise known as CESA8 proteins. Primary cell walls have a terminal complex composed of CESA proteins 1, 3, 6, and 9, while secondary cell walls have a terminal complex composed of CESA proteins 4, 7, and 8. In this experiment, we are focusing on CESA8 and its ability to synthesize cellulose in secondary cell walls. So from RNA quantification studies, we know that CESA8 is expressed in internodes, nodes, and young spikelets. Higher expression of CESA8 is seen in young spikelets as indicated by the orange coloring, but this expression ceases in a mature plant. In addition, CESA8 expression increases in internodes as the plant becomes more mature, but then decreases at some point in time. Thus, we come across one of the limitations of using RNA quantification to track gene expression. In this case, there are no samples between 35 days after germination and 60 days after germination, and there are absolutely no RNA samples after 60 days after germination, leaving us with big gaps in CESA8's expression throughout the plant's life. One way to combat this is through the use of a luminescence-based reporter system, such as luciferase. Luciferases are enzymes that catalyze reactions resulting in the emission of light from substrate, creating luminescence. The most commonly used luciferase in research comes from the firefly. Firefly luciferase catalyzes the emission of green photons from its natural substrate luciferin. The firefly luciferase cleaves the luciferin in the presence of ATP and oxygen to produce oxyluciferin, emitting luminescence as a byproduct. With an enzyme turnover rate of about 3 hours, firefly luciferase does not accumulate intracellularly, allowing for real-time measurements of gene expression. The way we're using the system is by taking a promoter from a gene of interest, placing it upstream of the luciferase coding sequence, otherwise known as LUC2O, and transforming this construct into brachy. We can then use these images. We can then image these transgenic plants using an extremely sensitive charge-coupled device camera that converts photons into pixels. So to really quickly go over vector construction transformation process in more detail, we started off with a plasmid received from the CEPRIC lab that contained the BD CESA8 promoter upstream of the gateway recombination sites. We then gateway cloned in the luciferase coding sequence, LUC2O, so that was downstream of the BD CESA8 promoter. We then transformed this expression construct into brachy using agrobacterium-mediated transformation. From here, we were able to regenerate multiple transformation events and grow other progenies to create BD CESA8 LUC2O plants. So these transgenic plants can be used in a myriad of ways, but for this experiment, I'm using them to track the spatiotemporal expression of CESA8 and therefore the secondary cell wall synthesis throughout the plant's life. Since emerging, these 8 CESA8 LUC plants have been imaged every single day, twice a day. So the first image comes between hours 8 and 10 of a 24-hour cycle, and this occurs during the light period where the temperature is 26 degrees Celsius. The second image comes during the dark period where the temperature is 18 degrees Celsius. And at every single time point, the image exposure time is always 20 minutes. And right here, we have an example of one of the racks shown used for imaging. So from these images, we were able to compile this list, and this video basically just shows what we have come up with so far. I'm going to slow it down so it's a bit easier to see CESA8 expression throughout. So these are when the plants are very young and still growing. We can see that we have some CESA8 expression, but as they start to grow more leaves and really elongate their stems, especially in this one, we see that CESA8 increases, and we can see it actually in the spikelets and in the internodes. So I know that that was really quick, but we have these screen caps here where we can more clearly see it. So the conclusions that we can draw from this is that our luciferase reporter system for CESA8 mainly supports the EFP browser data. So more specifically, we can see that the young spikelets or the younger spikelets have high CESA8 expression, and that CESA8 expression really decreases the more mature the spikelets get. But we can also see that CESA8 is expressed in the nodes, in the inner nodes where there's some expression, and also in the spikelets. One other thing to note is that CESA8 expression seems to be localized only at the bottom of the inner nodes and not at the top, which could be the result of secondary cell walls not being expressed at the top of inner nodes. And this bright field picture, this third picture right here, is to show the parts of the plant that may not be luminescent, so where CESA8 is not being expressed, but just so that you can get an overall structure of the plant. So some of the takeaways that we want to draw on from this experiment is we have this qualitative analysis showing that we think expression decreases the more mature plants get, but we can't know this for sure until we quantify it. So the next step in this process would be to quantify the luminescence using ImageJ. So there are 3 ways that we can do this, and the ways that we're most interested in are comparing light versus dark time points. Currently, we're struggling with quantification and comparison of light versus dark time points. Is as you can see in this light time point image, there's a lot more background noise as compared to the dark one, and that's making it very difficult to distinguish significant CESA8 expression from non-significant, just general noise in the background. So right now, all we have that we can quantify from right now is the dark time points. Another thing that we want to do in the future is to compare tissues of interest. So I've pointed out 3 tissues that we're specifically interested in where CESA8 has shown to be expressed, and that's the spikelets, the nodes, and the inner nodes. And we would like to compare these tissues of interest to see if CESA8 is more highly expressed in one tissue type than in another tissue type. In addition, lastly, we want to compare the same tissue over different developmental stages. So comparing spikelets when they're really young versus when they're really mature, and seeing how CESA8 expression changes would be very helpful. And seeing CESA8 expression over the lifetime of these transgenic plants. But for now, we are very happy with the qualitative data, but we really do hope to draw more from the quantitative analysis. Thank you for listening to my talk.